In our last class, we have started the chapter that is cell, the fundamental unit of life, and we have discussed briefly about the cell history, about the cell theory, and the differences between the types of cell, that is prokaryotic cell, eukaryotic cell, that is animal cell, and plant cell. Now today we are going to discuss in detail the structure of prokaryotic cell. So this is the prokaryotic cell in front of you, that is the bacterial cell. Now you can see the outer membranes. The outer membrane of the bacterial cell has three parts: capsule, cell wall, and the plasma membrane. Generally the capsule is made up of glycocalyx. The glycocalyx protect the cell, and this glycocalyx differs in the composition. If this glycocalyx is thick and strong, then it results in the formation of capsule. If it is a loose one, then it is known as the slime layer. Now this glycocalyx is related to the staining of the bacterial cell. In case of slime layer, that is the loose sheath, the stain is not taken up, so the bacteria becomes gram negative. Now if the glycocalyx is thick like a capsule, then it take up the stain or the color, and in that condition the bacteria becomes gram positive. Besides that, you can see the cell wall, which is providing the shape and rigidity. Then next part is the cell membrane. This membrane is semi-permeable in nature, and it allows the movement of material from inside to outside and outside to inside. So these are the three main layers that the bacterial cell wall is formed of, that is capsule, cell wall, and plasma membrane. The next very prominent structure that you can see is flagella. Now these flagella are basically helping the bacteria to move. The flagella is nothing but the extension of the cell wall, and it is formed of three parts, that is filament, hook, and basal body. Now you can see the small dot like structures which are known as ribosomes. The ribosomes are basically responsible for protein synthesis. In the bacterial cell or the prokaryotic cell, the 70S type of ribosomes are present, and these 70S type of ribosomes are made up of two subunits, that is 50S and 30S. The 30S is the smaller unit, and 50S is the larger unit of the ribosome. The S stands for the sedimentation coefficient. After that, you can see the mesosomes. The green color structure is there, that is mesosomes. These mesosomes are nothing but the infolding of the cell membrane. These mesosomes help in respiration, replication, cell formation. They increases the surface area of plasma membrane. Besides that, in certain prokaryotic forms like blue-green alga or cyanobacteria, the color pigments are present like chlorophyll, which are known as chromatophores. Now you can see the purple color round structures which are known as plasmids. These plasmids are extra chromosomal circular DNA material. These plasmids have great utility in genetic engineering, which you will learn in detail in your next class, that is the 12th standard. Besides that, you can see the jelly-like material which is labeled as the cytoplasm, and the pink color thread, entangled thread-like structure, which is the nucleoid. Now we have already discussed the nucleoid, that it is the region of cytoplasm where the genetic material, that is DNA, is scattered. So this is a brief or a detailed account about the structure of the prokaryotic cells. So these are the new topics that you are learning. You are learning about the capsule, you are learning about mesosomes, you are learning about the nucleoid, right? So this is all about the structure of the cell, that is the prokaryotic cell or primitive cell, where the only the ribosome as the cell organelle is present. The other cell organelles like mitochondria, endoplasmic reticulum, they are totally absent in these type of cells. Now let us see the structure of eukaryotic cells. This is the structure of eukaryotic cell with well-defined cell membrane, plasma membrane, cell wall, then all the cell organelles you can see here. This is the plant cell shown. So you can see the chloroplast also, you can see Golgi also, you can see nucleus also, rough and smooth endoplasmic reticulum, a prominent nuclei can be visualized. So these are the variety of cell organelles that are present in the cell. Now in the eukaryotic cell we generally discuss the cell envelopes, then the cytoplasm, and finally the organelles one after the other. So we are going to study about the endomembranous system first. Now the endomembranous system, as the name suggests, that it is present within the cell and it is consisting of membranous organelles. So the endomembranous system is formed of four cell organelles, that is endoplasmic reticulum, Golgi body, vacuoles and lysosomes. These four organelles are forming the system. That system is known as endomembranous system. This endomembranous system is basically helping in transportation and manufacture of material from one part of the cell to another part. Now we will discuss the endoplasmic reticulum first. The endoplasmic reticulum is of two types, RER and SER. RER means rough endoplasmic reticulum and SER means the smooth endoplasmic reticulum. These endoplasmic reticulums are basically responsible for synthesis of proteins and lipids, and these proteins and lipids are further helping in formation of the cell membrane, and so this is known as membrane biogenesis. Besides that, they also help in detoxification of the cell. The next organelle that is the part of endomembranous system is the vacuole. Now the vacuoles are the round or spherical bodies or the oval bodies, and their main function is to store the material. On the basis of the material they store, the vacuoles are different types, like food vacuole is there, like excretory vacuole is there, then contractile vacuole is there. So depending upon the content within the vacuole, the naming of the vacuole is given. After that, the next organelle is the lysosome. The lysosome is known as the digestive bags. Now they are known as digestive bags because they have got several digestive enzymes in them, and these digestive enzymes help in intracellular digestion, and they also kill the foreign body if it enters the cell by the process of phagocytosis. There are various enzymes present in lysosomes that help in the digestion of protein, carbohydrate and lipids. Now besides this, the lysosomes are also called suicidal bags. They are known as suicidal bags because if the cell organelle is not working properly, or if the cell is not working properly, then these lysosome burst, release their content in the cytoplasm, and that content digests the whole cell. That is, it compels the cell to do the process of suicide. That is why it is known as suicidal bag. Now the last individual or part of the endomembranous system is the Golgi body. Camillo Golgi was the one who discovered the Golgi bodies, and these Golgi bodies are the membranous structures with the flat disc-like structures that are known as cisternae, and the round ball-like structures which are known as vesicles. Now they are basically involved in packaging and transport of material. Whatever protein and lipids are synthesized by endoplasmic reticulum, they are modified by the Golgi before it is released. They also help in the formation of lysosomes. They also help in the formation of pigment like retinal and melanin. They also help in formation of ATP or synthesis of ATP. So these are the main functions that are performed by the Golgi bodies. Now the Golgi body synthesize the lysosomes, and those lysosomes are labeled as GERL, GERL. Now when I expand GERL, it means Golgi Endoplasmic Reticulum lysosomes. So here we have discussed the 4 types of organelles. In your lower classes you have discussed these organelles as a separate one, but here we have discussed these 4 organelles under a single heading, that is the endomembranous system. That's all for the day. Thank you.